Okay, so we are very quickly going to go over taste. So for taste, um, let's see, we have our basal cell, which is something, so this is basically going to be representing our basal cell. And actually, so this is our this is our taste bud. So this would be our taste bud. And each taste bud, this has around 50 to 150 taste receptor cells that are sandwiched in between like supporting cells and other basal cells. And then the receptor cells are specialized epithelial cells that synapse. And then we have our receptor cell, yeah, they synapse with neurons. So that's what this is. So this is our receptor cell, sorry. Our receptor cell that is specialized epithelial cells that synapse with neurons that can basically excite the neuron when it interacts with a taste stimulus. So our taste stimulus would happen up here. Um, yeah, so our tastants react with the receptors. Salt would be sodium, sour is potassium, and then bitter, sweet, and umami deal with G-coupled protein receptors. So, yeah, each cell receptor cell is orientated in the receptor taste bud that protrudes into the oral cavity through a taste pore. So this right here is going to be our apical surface where the tasting is dissolved. And then this is our apical end where it responds to the tastant here being dissolved. And then this is our basal end, which connects to our neuron. And the basal end transmits signals to the brain. So let's start with sodium. So we have something called receptor potentials, which is when the depolarization of a non-nerve cell reaches its graded potential, which modulates the activity of ion channels to trigger an action potential. And the resting membrane potential for this would be around negative 70 millivolts, similar to our um, neuromuscular junction. So how this works is that we have a neurotransmitter. The neurotransmitter is basically going to be released out here. We don't know what the neurotransmitter is. That's the thing. So we just say neurotransmitter is released from the basal membrane of the taste receptor cell, and then when the tastin interacts with the apical surface of the receptor cell, it interacts and creates a depolarizing receptor potential, and this is going to cause the opening of like calcium channels on the basal surface, which technically would be like down here, but for our purposes, it's right here, and then it just connects down here. Um, yeah, so the substance is dissolved in the saliva up here, and then it interacts with the apical membrane of the cell, and then it increases the movement of positive charged ions across the membrane. So we have for saltiness, sodium. Sodium's going to go ahead and enter the cell through these ENaC channels, which is electrically gated sodium calcium channels. So the increase of sodium, which is a positive charge going into the cell across the membrane, is causing a depolarization, and then this depolarization is going to cause calcium to come in, and then calcium is going to bind the snare proteins, and then cause exocytosis of this neurotransmitter. Which is then going to further rise depolarization of this neuron, which is going to increase intracellular calcium. And then the same for sour. Sour uses an ENaC channel, but we have hydrogen going on causing depolarization just because of the positive charge. And then calcium is being triggered by this depolarization, which is then going to bind to snare proteins and then release neurotransmitters via exocytosis to the neuron, which is going to cause another depolarization and then increase in intracellular. The same thing is going to occur over here with the sweet, bitter, and umami, but the only difference is that so we use G-coupled protein receptors, but they don't cross the membrane. So Yeah, so they don't cross the membrane, but the effect is still going to be the same for them. And then once they do bind to these G-coupled protein receptors, it triggers basically a second messenger, which causes the opening of calcium, but it's going to be over here rather than over here. So triggers the opening of calcium on like the surface of the taste receptor. So it could be over here, or it could be in the smooth endoplasmic reticulum, which is kind of like what our book showed. And then calcium enters the cytosol, and then that triggers exocytosis of the vesicles, full of the neurotransmitter, and then yeah, it goes to the neuron, it dissolves in there, and then we get our action potential. That's received from different receptor types. And then as for smell, it's not similar in process, but action potentials and everything like that. So what's going to happen is that the olfactory epithelium is basically in like the roof of your canal. So say this is the nose, and then this is the epithelium. That is going to be, so the odorant is going to be dissolved in there, in the mucus layer, and I think that it being dissolved in the mucus layer helps because you can absorb the smell, and then you create an action potential. So, the dissolved odorant binds to the receptor located within the membrane of the receptor cilia. So this is our little cilia right here, of the olfactory neuron, and then so these G-coupled protein receptors activate adenylyl cyclase, and then that converts it into cyclic AMP, and then this allows for so this whole process right here, going from adenylyl cyclase to CAMP, is going to cause the opening of sodium and calcium channels so that they can flow into the cell. And then this, like, all these positive charges are going to go ahead and create a depolarization of this neuron. And then we get our action potential, and then the action potential, this little thing right here, our action potential is going to cause these chloride channels to open, and then chloride, look at this, is going to leave the cell, making it even more positive. Yeah, so our olfactory neurons synapse with neurons in the olfactory bulb that go into the olfactory cortex, and then it has two options of like where it could go. So it could go to the temporal lobe, or it could go to the frontal. And our frontal we can think of as being emotions, and then temporal would be where it's perceived. And then smell doesn't pass through the thalamus. It's considered a primitive sense, which hormones can enhance cortisol regions of the smell and increase sensitivity.
Okay, so the way that we're able to focus on light is through mostly the lens. So, what we have here. Oh, wait, I want to make sure the audio's on. Okay, thank goodness. Okay, so we have our cornea, or I guess let's go over here. Actually, no, we're going to stay over here. So right here we have our cornea, and then we're just going to say that this is our lens, but it's going to be shown a little bit different right here. So this is our lens right here. And then we have these suspensory ligaments, which don't have any, so there's no tension on the suspensory ligaments. But when there is tension, the lens thickens. So it's at its natural shape. So tension equals thickens. So tension, oops. Tension on suspensory ligaments is equal to increase in thickness of the lens. And then our ciliary muscles, when they relax, tension on the suspensory ligaments increases, and then the lens flattens. So tension on the sensory ligaments equals an increase. Wait, now I'm confused. redo this really quickly. Okay, so we're gonna talk about how the eye focuses on light. So right here we have our cornea and then we have our lens, but it's going to be shown over here that this is going to be the lens. So the lens is basically right here. That's what I'm trying to point at. And then these are the suspensory ligaments. And then around here would be our ciliary muscle. This is the fovea, and this is where light is basically transmitted and where we see the most amount of focus being sent to the brain. And so our suspensory ligaments, they're surrounded, so they are surrounded by elastic lens attached to the ciliary muscles via the suspensory ligaments. Oh my gosh. Okay, so lens is attached to the ciliary muscle through the suspensory ligaments. And then when the suspensory ligaments contract, that means our lens tends to thicken. And then when they flatten, there's less tension right here, so they're able to flatten. Oh my gosh, let me rephrase this. So when there's no tension on the suspensory ligaments, that means the lens is at its thickest or its natural shape. I can't talk right now. So the ciliary muscle, when it's relaxed, tension on the suspensory ligaments increases and then the lens flattens. So relaxation, not that this is the most important thing right now. So relaxation of the lens is equal to tension on suspensory ligaments, which causes them to increase, and the lens flattens. And then we have, so we also have our anterior cavity, which is the space between the lens and the cornea. This one is where we produce our aqueous. Aqueous humor would be over here. Yeah, aqueous humor would be over here. And then we have our vitreous humor over here. So the way focusing on the retina, so focusing light on the retina is the most important. So light has to be reflected off of an object. So light has to be reflected off of an object in order for us to see it. And then, so it has to be bent, which is kind of like what we're showing right here, this little kink right here. It has to be bent or refracted. And then the convex structures, or the cornea and the lens right here, these two are going to bend the light, so then it's transmitted over here to our fovea. And depending on how close the object is, the light will diverge at different angles. So closer, let's see, okay, so basically the closer that the object is, the more light has to diverge and it has to be refracted to focus on the cornea, which is required for most of the bending, and that can't be adjusted. So right here, this is our focal point, and then we have our optic nerve that basically sends information to the brain. So the focal point right here, the fovea, this is a precise point at which all rays of light converge at. Okay, so the thicker the lens, so thicker lens equals a shorter focal point, which means that we're able to bend light more sharply, so like your acuity, which means we're able to bend light more sharply. And then a flattened lens, which is basically when our suspensory ligaments are increased in tension, that's when they flatten. This is when we have a longer focal point, which equals us to be able to bend light at a lesser degree. So the far point of vision is kind of like the closest distance of which the lens doesn't have to thicken in order for it to properly focus on the object. So the lens, it can see normally at 20 feet, but some people have, so there's a process in far point vision that we have to take into account where one, we have accommodation where the lens has to thicken, and then two, so the lens, it has to thicken in order for light to be able to shine through. And then this is where we converge, which would be on the lens, which is where the light will converge, and then the pupils will either constrict or they'll dilate. Sorry. So the pupils will constrict, and then that'll increase the depth of focus and like how much of the visual field we can keep in sharp focus. But there are some focusing errors. Okay, we don't need to talk about that. We just need to describe the two types of photoreceptors and explain how they convert light into electrical energy, and explain how the center and surround incorporates in visual acuity. Okay, so we're gonna actually go over here to this amazing little chart that I have. So, photoreceptors. This is our photoreceptor here. And yeah, so this is our little photoreceptor right here. And then we have our bipolar cell. And then this is our ganglia. So this is kind of like, so this whole thing right here is going to be our, like, so this is the photoreceptor right here, and then this is the back of the retina. And in the back of the retina, we have our 11 cis retinal, and our 11 are all trans retinal. So right here, we have this little G coupled protein receptor right here, and we have like our alpha subunit, which we're going to say is right here, but this is like a beta, gamma, but transducin is referred to as our alpha subunit. And then here is when we have an action potential where calcium is going to come into the cell, and we're going to cycle cGMP, and then calcium is going to cause an exocytosis of these neurotransmitters, which are known as glutamate, into this little area, which is going to allow sodium in as well. And then these, so one thing to know is less, so this is actually going to be in less glutamate, yeah, so less glutamate is going to, so this is going to be in the dark, and when we have glutamate, it's going to bind to this glutamate, which has these three little things, and then the binding of glutamate to here is going to cause TRPM1 to open, and then that's going to allow sodium or potassium into the cell. So sodium's going in, and then potassium is going out. There we go. And then with this as well, we have our cGMP, which is right here, which we kind of had right there. This is going to cause calcium to come in, which is then going to cause it to bind to snare proteins and then release glutamate. So this is also our glutamate, and then that is going to depolarize the cell, and that's going to cause these AMPA channels open, which are sodium channels, but they only respond to glutamate. And then that's going to cause an action potential, and it's going to send its signals to the brain. Oh, I don't know why I didn't include this. Sorry, I'm looking through my notes of what I was going to say with all this. Yeah, so we have our cones, which are responsible for our, so our cones are going to be responsible for color vision and visual acuity. And then our rods help with low light and peripheral vision. They don't detect color. We could say that they are, they only see in black and white. And then people who have kind of like an elongated eye, this is when we need to be able to converge light more, because they won't see anything until further back, until it hits the fovea right there. So the light will converge here. So this one is going to be referred to as our concave lens, and then this one is our convex. And convex is going to be used when people who have nearsightedness. We can associate that nearsightedness. And then convex we can think of as farsightedness. So yeah, okay, so with concave lenses, what they're gonna do is they're gonna be able to help us spread light more, to lengthen the focal point. Because it's too short. Because normally it's right here, but it's going to help lengthen it to this point just because of how the light is being converged. And then our convex lens, which is used for people who are farsighted, that is when your eyeball is too short. It normally, reaches here. This one is when it's too long, but this one is when it's too short. And the object tends to fall behind the retina instead of in front of it. So the convex lens helps to shorten the focal point because it's normally at here, but we want to get it to right here. So that's something I should have mentioned in the first place. Yeah, so phototransduction. So here is when we're in our unexcited state. So we could say that this is our unexcited state. And we have rhodopsin, which is basically this visual pigment that's composed of opsin, which is a protein. And it also has retinol in it, which is derived from vitamin A. So in its unexcited state, it fits in the binding site of opsin. So this fits right here in this nice little part. But when the wavelength is absorbed, so when light comes in and is absorbed by rhodopsin, sorry, I don't know why I'm losing my mind. So the cis transretinal becomes an all transretinal, and basically like unkinks right here, and it still has this double bond, and it no longer fits in this binding site right here. It can't fit anymore. So rhodopsin ends up detaching from opsin, which would be our transducin. Retinol becomes inactive, so it becomes inactive, and then it associates with the opening of opsin until it regains its original conformation. So the GPCR on the photoreceptor, so this is our GPCR right here. Yeah, It activates the protein with the alpha subunit, and then the alpha subunit brings a change of, so the alpha subunit brings a change of transducin, which allows ions to flow in, which creates this positive charge, and then calcium comes in. We get that, and that's when we're in the dark. So when we're in the light, though, transducin is going to be activated. So right here, transducin, they're activated, which means that this alpha subunit right here is going to activate phosphodiesterase, which would be right here. So this is when we're in light, right? Okay, yeah, so we have our phosphodiesterase. So this is when we're in light. And then this enzyme, okay. So phosphodiesterase, this little enzyme right here is going to break down cGMP into cyclic AMP. So into GMP, and then that's going to cause the opening of cation channels. So a cation channel like sodium's going to come in, and then that's going to cause it closes the channels, and then it hyperpolarizes. So these channels, they become closed, and then we hyperpolarize, taking it more negative because we're not allowing any positive ions to come into the cell, which then removes our inhibitory response. So when this is closed, nothing happens. So there is no release of glutamate, and then the removal of this inhibitory signal to the bipolar neuron allows them to fire this action potential, because we have this here. We're not our glutamate or CAMP isn't going in. This is also closed. This doesn't exist, and it turns into our AMP, and then we still don't have this. But because we don't have this, that's going to cause the release of glutamate, and then rhodopsin diffuses into the pigment epithelium where the enzymes act to restore the 11-cis-2 state right here. And then it goes in and it goes to the brain. So when you have more glutamate, that means you are going to have less glutamate. You can still have two action potentials no matter what's happening. Yeah. So I don't think I mentioned this, but this is our mGluR6, our metatropic glutamate. So it responds to glutamate ligand, but work through a second messenger that causes the inhibition of calcium. So the inhibition of calcium, which is the TPRM1 channels, and then the bipolar turns on when glutamate releases. So we can think of it as this. So on is equal to when glutamate is released. On is glutamate release in light. And then off would be an increase in glutamate. So less glutamate, increase in glutamate. And this is in the dark. Because in the light right here, no, I was right. Okay. So yeah, right here, because we have no glutamate release, there's not supposed to be any right here. These close and they just don't open again. And then we have but when this does happen, we have these so we can think of it, so photoreceptor, this is a photoreceptor, but right here, this would be our surround, and then this is our center. So we still have our transducin, which is our alpha subunit, and our G-coupled protein receptors, the phosphodiesterase, and then our channels right here as well. And then, so, calcium, it'll come in creating a positive charge within the cell, and then that'll cause the binding to snare proteins, and then it'll cause an exocytosis of this neurotransmitter GABA, and instead of glutamate, we're using GABA, which then binds to this horizontal cell, which uses AMPA channels. And this, it's only activated in the dark. So maybe I did switch them. Okay. So in the dark, we can think of it like there's an increase in glutamate release, kind of with the first one too, which will inhibit this center, which causes us to not release glutamate, which then closes down this channel, but then that can come in, and then we cause that action potential there. But in the light, it's inactive, and then there is no, because there's no this, there is no glutamate release, which then causes the action potential. So when it depolarizes, the opposite of this cell releases the neurotransmitter GABA onto the terminal end of the center of the photoreceptor, and then that acts to inhibit the release of glutamate from the photoreceptor, and then the decrease in glutamate to inhibit the glutamate from, it inhibits, so GABA inhibits the glutamate release from the photoreceptor, and then the decrease in glutamate from the photoreceptor causes a decrease in glutamate going from the receptor to the bipolar neuron. Actually not confident in this at all. So we have our ear, then we have our cochlear duct, and then we have these parts of the ear which help transduct the vibrations that we hear to the cochlea. And the cochlea, it's actually kind of like this spiral, but for the sake of this, it's just easier to draw it like that. So we have our malleus right here, our incus right here, and then our stapes, which connect to the scala. So this is our scala tympani, scala, oh no, hold on, what is it? It's called the scala, not tympani, it's something else, I can't think right now. I don't know, but yeah, and then right here we have this little middle part, which is the cochlear duct, and this one has endolymph in it, and the scala tympani and the other scala right here, these have our perilymph. Yeah, so it's our scala media, or the cochlea duct. And then, so sound, what it's going to do, it's going to enter, and then it's going to strike the tympanic membrane, which is basically going to be this whole thing right here, or your eardrum, and then it's going to cause it to like vibrate. And then in the middle of the ear, so I guess, yeah, right here in the middle of the ear, the vibrations are transmitted through the ossicles. So this right here would be our tympanic membrane, and then it goes to our ossicles, and then these basically amplify the sound and then convey it to our oval window of the cochlea. And then we have this inner fluid of the stapes that move the oval window to kind of create this clear fluid that leads to the motion of the basilar membrane within the cochlea. So this would be like our basilar membrane. So what's happening here is that it has like, so this would be like our little hair follicle. Yeah, so this is like our like little hair cell activation. So the movement of the basilar membrane right here is going to cause a deflection in the organ of the cordae. And then it's like a deflection of opening of channels. So we have our voltage gated calcium channels, which binds to this, and then we go up here, and then this is Preston, and I'll get into that in a second because it deals with chloride channels. Yeah. So, so calcium, it causes this kind of like wave. So we have these channels, and then we have like these little springs that are attached to each one of these, and when you like pull them together, it's gonna start to move towards here, and then these opening of channels basically causes a depolarization, and then this is where like the electrical signals are amplified, and potassium channels open, which is causing more positive charges to enter the cell and depolarization. And then Preston, which has voltage gated calcium chloride channels, this they were talking about it in class and like in tutoring, how just like through evolution they just like stopped working, so it doesn't open. So these are just kind of like hanging out right there, but what it'll do is it'll shorten here, like this will not sarcomere, shorten this because of the change in voltage. So we have the inner ear, so the outer ear regulates the pumping, which causes us to hear. But right here we have our tectorial membrane, and this is kind of like where the signal transmission of action potentials kind of goes through. So it goes through the auditory nerve to the brain, where it's interpreted as sound. So we're basically, what we're doing is we're blowing this up, and that's coming to here. So we have our temporal membrane, and then when these start to like kind of move, because remember how they're like connected through each these are connected through each little thing. So when the action potential comes through, this starts to move, and these kind of like start to cock towards here, if that makes sense. And then it'll be opening the sodium and potassium pump, and then that is through our active transport. And the resting membrane potential of this is around -40. The scalea tympanic membrane, which again is right here, has perilymph, and the stria. And then once we kind of get this going right here, this little movement, I don't know what it's called exactly, kind of maybe like a cross bridge formation, it's going to send its signal to the brain. And then we have in the endolymph a high driving force of potassium, which is around 120 millivolts, because potassium's going in. We just have a bunch of positive charges, so this chloride channel has nothing to do with Preston. It's just going in, but there's no energy required. So this is our endolymph, and then here we have our potassium leak channels. Those are over here as well, so they're going to be on the opposite side of the pump. So you can see like right here we have a division between the two. The sodium's going out, potassium's going in. Potassium has a higher driving force than it does for both sodium and chloride, and then it has the blood, which then causes these like little vibrations. So the sound comes from the temporal lobe, and then the sensorineural neural is essential caused by a stroke. So maybe that wasn't too bad.